The human brain, an organ of unparalleled complexity and energy consumption, undergoes a silent but profound physiological transition as we cross the threshold of the fifth decade of life, a shift that is often imperceptible in its early stages but cumulatively devastating if left unaddressed. For the individual over the age of 50, the primary concern is no longer merely the acquisition of new knowledge, but the preservation of the cognitive architecture that houses a lifetime of memories, skills, and personality, a structure that is increasingly besieged by the biological forces of aging. To understand how we can aim for a theoretical 100% improvement or restoration of memory function, we must first confront the cellular reality of the aging brain, specifically the dual threats of oxidative stress and neuroinflammation, often referred to collectively as inflammaging. As metabolism slows and the body's endogenous antioxidant defense systems, such as glutathione and superoxide dismutase, begin to decline in efficiency, the brain becomes disproportionately vulnerable to free radical damage. Weighing only about 2% of total body weight, yet consuming 20% of the body's oxygen, the brain is a high-performance engine that generates massive amounts of metabolic exhaust. When the cleanup mechanisms falter after 50, this exhaust begins to rust the machinery of thought itself, particularly in the hippocampus, the seahorse-shaped region responsible for the consolidation of short-term memory into long-term storage. This oxidative corrosion leads to synaptic pruning, where the connections between neurons wither like drying roots, and eventually to neuronal apoptosis, the death of the cells themselves. However, the prevailing medical dogma that neuronal loss is irreversible has been overturned by the discovery of adult neurogenesis, the brain's ability to grow new neurons throughout life, provided the biological environment is conducive. It is within this window of opportunity that specific bioactive compounds found in berries, particularly a class of flavonoids known as anthocyanins, emerge not merely as food, but as potent pharmacological agents capable of crossing the blood-brain barrier to trigger genetic switches that govern cellular repair and regrowth. The first and perhaps most scientifically substantiated pillar of this regenerative quartet is the blueberry, specifically the wild variety Vaccinium angustifolium, which stands as the gold standard in neuronutritional research due to its extraordinarily high concentration of anthocyanins, the pigments that give the fruit its deep blue-purple hue. While the layperson sees a fruit, the neurobiologist sees a delivery system for delphinidin, malvidin, and petunidin, specific molecular keys that unlock the brain's potential for repair. When a person over 50 consumes blueberries, these compounds are not simply digested for energy. They undergo a complex metabolic transformation in the gut, entering the bloodstream and, crucially, possessing the unique ability to traverse the selective permeability of the blood-brain barrier. Once inside the neural environment, they do not act as simple antioxidants, a common misconception, but rather as signaling molecules that interact directly with the ERK/CREB pathway, extracellular signal-regulated kinase/AMP response element binding protein. This interaction is pivotal because the CREB protein functions as a transcription factor that activates the genes responsible for the production of brain-derived neurotrophic factor, or BDNF. In the landscape of geriatric neuroscience, BDNF is widely regarded as miracle grow for the brain. It is the essential protein that encourages the growth of new neurons in the dentate gyrus of the hippocampus and strengthens the synaptic plasticity of existing connections. A deficiency in BDNF is a hallmark of age-related cognitive decline and Alzheimer's pathology. Yet studies have consistently demonstrated that consistent supplementation with blueberry polyphenol can elevate BDNF levels, effectively shifting the brain from a state of degeneration to one of regeneration. This mechanism of action extends beyond direct neuronal stimulation to the critical support system of the brain, the cerebrovascular network. As we age, the microvessels that feed oxygen and nutrients to our neurons tend to stiffen and narrow, a condition exacerbated by endothelial dysfunction where the lining of the blood vessels loses its ability to produce nitric oxide, the gas responsible for relaxation and dilation. This reduction in cerebral perfusion means that even if neurons are healthy, they're starving for fuel, leading to a brownout effect where cognitive processing speed slows and memory recall becomes sluggish. The anthocyanins found in blueberries target the endothelial cells of the cerebrovascular system, upregulating the activity of endothelial nitric oxide synthase, ENOS. This enzymatic activation results in improved vasodilation and a measurable increase in blood flow to the gray matter, particularly during cognitive tasks. Furthermore, we must address the issue of microglial activation, a topic that has moved to the forefront of neurodegenerative research in the last decade. Microglia are the immune cells of the central nervous system, functioning as the brain's janitors, constantly patrolling for pathogens and damaged cells. In a healthy, youthful brain, they're quiescent and helpful. However, after the age of 50, largely due to systemic inflammation and accumulated metabolic debris, these cells can become chronically activated, shifting into a primed state where they become hyperaggressive, attacking healthy synapses and releasing inflammatory cytokines. The polyphenolic compounds in blueberries have been shown to exert a potent calming effect on microglia, modulating their phenotype from the pro-inflammatory M1 state to the anti-inflammatory and restorative M2 state. Thus, the blueberry acts as the architect of our neural recovery plan, laying the structural groundwork for neurogenesis, while the blueberry functions as the benevolent architect of the brain, laying the foundation for neurogenesis and establishing the vascular irrigation necessary for growth. The restoration of a 50-year-old brain requires more than just new construction. It necessitates a rigorous demolition of the dilapidated cellular structures that clutter the neural landscape, a process that brings us to the second and often underestimated powerhouse of the berry kingdom, the strawberry. In the hierarchy of nutritional neuroscience, the strawberry has frequently been dismissed as merely a vessel for vitamin C and sugar, yet recent advancements in gerontology have elevated it to a position of supreme importance due to its unique concentration of a bioactive flavonoid called fisetin. To understand why fisetin is the critical second key to memory restoration, we must confront the phenomenon of cellular senescence, arguably one of the most detrimental hallmarks of aging tissues. As neurons and glial cells age, they do not always die and undergo clearance via apoptosis. Instead, many enter a state of suspended animation known as senescence. These zombie cells cease to divide or function functionally, yet they remain metabolically active, occupying vital space within the hippocampus and, more dangerously, secreting a toxic cocktail of pro-inflammatory cytokines, chemokines, and proteases known collectively as the senescence-associated secretory phenotype, or SASP. This SASP acts like a contagion, poisoning neighboring healthy neurons and spreading inflammation in a widening circle of dysfunction that actively inhibits the formation of new memories. Herein lies the brilliance of the strawberry-derived fisetin. It is a potent senolytic agent, a compound capable of distinguishing between a healthy cell and a senescent one, selectively inducing apoptosis in the latter to clear the dead wood from the forest of the mind. The consumption of strawberries, therefore, is not a passive nutritional act, but an active intervention in cellular quality control. When fisetin enters the neural environment, it modulates the PI3K/AKT/mTOR signaling pathways, which are central to cell survival and growth. By inhibiting the pro-survival signals in these damaged, senescent cells, fisetin effectively strips them of their defense mechanisms, forcing them to self-destruct and be cleared away by the immune system. This taking out the trash is a prerequisite for the efficacy of the neurogenesis stimulated by blueberries. Simply growing new neurons in a toxic environment filled with CSP-secreting zombie cells is futile, as the new cells will likely perish or fail to integrate into existing circuits. Furthermore, fisetin has been shown to directly enhance long-term potentiation, LTP, the biophysical process by which synaptic connections are strengthened based on recent patterns of activity, essentially the physiological basis of learning and memory. By clearing the inflammatory interference and directly stimulating the signaling pathways involved in synaptic plasticity, the strawberry acts as the clarifier of the cognitive signal. However, the restoration of memory is not solely a matter of static data storage. It is intrinsically linked to the dynamic execution of movement and coordination, leading us to the third vital component of our regenerative quartet, the BlackBerry. Clinical neurology tells us that a decline in gait speed and balance is frequently one of the earliest predictors of cognitive decline. The BlackBerry, with its unique subclass of anthocyanins, primarily cyanidin, 3-glucoside, possesses a unique affinity for the motor control centers of the brain, protecting the striatum and cerebellum from oxidative damage and preserving the integrity of the motor-cognitive interface. The mechanism by which blackberries exert this protective effect extends deep into the mitochondria, the energy powerhouses of the cell. Neurons are obligate aerobes with a voracious appetite for energy. When mitochondrial efficiency drops, a condition known as mitochondrial dysfunction, the neuron lacks the ADP, adenosine triphosphate, required to maintain the ionic gradients necessary for firing action potentials. This energy crisis is particularly acute in the highly active motor neurons. Cyanidin, 3-glucoside found in blackberries, has been shown to stabilize the mitochondrial membrane potential and reduce the leakage of reactive oxygen species, ROS, during energy production. By plugging these leaks and optimizing the electron transport chain, the blackberry essentially tunes the engine of the brain, allowing it to run smoother and cooler. This mitochondrial support translates into a brain that is not only better at moving the body, but is also liberated from the constant background stress of motor inefficiency. Consequently, the cognitive reserve is freed up. The mental energy that was previously squandered on trying not to fall or stumble can now be reallocated to the processes of recall, problem-solving, and creative thought. Furthermore, the blackberry contains high levels of manganese, a trace mineral that is a critical cofactor for the enzyme manganese superoxide dismutase, and MNSOD, the primary antioxidant defender within the mitochondria itself. This internal defense system is crucial for preventing the rusting of the neural circuits over decades of use. Moreover, the synergy between the blackberry's polyphenol and the brain's inflammatory response offers another layer of protection. Research indicates that the specific ratios of phenolic acids in blackberries can suppress the expression of COX, two enzymes in the brain, which are responsible for producing pain and inflammation signals. Chronic low-grade inflammation in the cerebellum and brainstem can lead to a stiffness in both thought and movement. By dampening this inflammatory cascade, blackberries contribute to a fluidity of psychomotor speed, the speed at which the brain detects a stimulus and executes a response. In the context of 100% memory improvement, psychomotor speed is a critical component. It is the difference between having a memory on the tip of the tongue and being able to articulate it instantly. The blackberry ensures that the transmission lines between the archival storage of memory and the active execution of speech and movement remain open and high speed. It prevents the lag time that often frustrates the aging mind, bridging the gap between intention and action. Therefore, while the blueberry builds the neuron and the strawberry clears the cellular debris, the blackberry energizes the system and harmonizes the complex dance between thinking and doing, ensuring that the physical vessel of the body remains a competent vehicle for the mind. As we stabilize the physical substrate of the brain with these first three berries, we create a robust, energized, and clean neural environment. Yet, there remains one final frontier in our quest for complete restoration: the metabolic flexibility of the brain and the protection of the lipid membranes that encase every single thought we possess. Having established the regenerative architecture with the blueberry, the senolytic purification with the strawberry, and a mitochondrial revitalization with the blackberry, we arrive at the final pillar of our restorative quartet, a fruit often relegated to the status of a seasonal garnish, but which, in the realm of neurobiology, acts as the guardian of the gut-brain axis and the protector of the blood-brain barrier, the cranberry, Vaccinium macrocarpum. The inclusion of the cranberry is critical not merely for its antioxidant capacity, which is formidable, but for its unique content of A-type proanthocyanidins, a specific molecular structure that differs significantly from the B-type found in most other fruits. In the context of the aging brain, specifically after the age of 50, the integrity of the neurological system is increasingly compromised by pathogens and metabolic toxins that migrate from the gastrointestinal tract. This phenomenon, known as leaky gut, allows lipopolysaccharides, LPS, and other inflammatory bacterial byproducts to enter the systemic circulation, where they eventually assault the blood-brain barrier, BBB. Once this barrier is breached, the brain is subjected to a state of chronic immune activation that degrades memory and cognition. The cranberry functions as a non-stick agent, widely known for preventing bacteria from adhering to the urinary tract, but less known for its ability to prevent pathogenic adhesion in the gut lining and the vascular endothelium. By fortifying the gut barrier and preventing the translocation of toxins, the cranberry effectively seals the borders of the brain, creating a sanitized environment where the regenerative work of the other berries can proceed without constant interference from systemic infection. Furthermore, the cranberry plays a pivotal role in the maintenance of membrane fluidity and signal transduction. The neurons in our brain are encased in a lipid bilayer, a fat-based membrane that must remain fluid and flexible to allow neurotransmitters to dock efficiently at their receptor sites. As we age, the accumulation of cholesterol and the oxidation of lipids cause these membranes to stiffen, a process analogous to the hardening of arteries, which essentially locks the doors of the neurons, preventing them from receiving the chemical messages of memory and learning. Recent research suggests that the polyphenol in cranberries can interact with the lipid head groups of the neuronal membrane, protecting them from lipid peroxidation, the chemical process that turns fats rancid. By acting as a preservative for the brain's fatty structures, cranberries ensure that the receptors for acetylcholine and dopamine remain accessible and responsive. This is the lubrication of the cognitive engine. The synthesis of these four distinct biological actions, neurogenesis, blueberry, senolysis, strawberry, mitochondrial energy, blackberry, and barrier protection, cranberry, forms the basis of the 100% improvement protocol. It is imperative to understand that these fruits are not merely interchangeable snacks. They're distinct pharmacological tools that, when combined, create a synergistic effect far greater than the sum of their parts. The protocol for the aging individual, therefore, is not a suggestion of casual consumption, but a prescription for daily integration. The science indicates that consistency is the key variable. The half-life of these anthocyanins and flavonoids in the human bloodstream is relatively short, typically ranging from 2 to 4 hours. To maintain a constant state of neuroprotection and regeneration, the brain requires a pulsed delivery of these compounds. Practically, this translates to a neuroberry bowl strategy. The recommendation is a daily intake of approximately 1 cup, 150 grams, of a mixed blend of these 4 berries. The timing of this consumption also leverages our circadian biology. Consuming these berries in the morning, perhaps alongside a source of healthy fat like walnuts or yogurt, optimizes absorption, as many of these fat-soluble compounds require lipids for transport across the intestinal wall. Moreover, the metabolic boost provided by the blackberries aligns with the body's natural cortisol awakening response, priming the motor and cognitive systems for the day's demands. It is also crucial to address the form of the fruit. While fresh is often touted as best, in the case of berries, frozen is often superior for the purpose of consistent neuronutrition. Freezing berries immediately after harvest locks in the polyphenol content, which otherwise degrades rapidly at room temperature. Furthermore, the ice crystals formed during the freezing process rupture the plant cell walls, effectively predigesting the fruit and making the anthocyanins more bioavailable to the human body. However, the 100% improvement claim is not a promise of magic. It is a promise of biological potential. It is based on the premise that the average 50-year-old brain is functioning significantly below its design capacity due to the unmitigated burden of inflammation, oxidation, and cellular senescence. By removing these breaks and pressing the accelerator of neurogenesis. The subjective experience of the individual is a radical restoration of clarity. The path to a revitalized memory after 50 is not found in a futuristic pharmaceutical lab, but in the ancient, co-evolved chemistry of the plant kingdom. The blueberry builds the road, the strawberry clears the debris, the blackberry fuels the engine, and the cranberry secures the perimeter. It is time to eat with purpose, to age with defiance, and to reclaim the full potential of our cognitive heritage.